Yeah, I'm just trying to see if this is working or not. Hopefully it is. Let's see. Actually, can you pull up the live stream on your phone and just see? You said you could do that, right? You're not. I mean, it's a public network. Oh, actually, you know what? I can do it myself. Don't I? I forgot. My phone is not logged in. I can just check.
Yeah, I see it.
Okay, good. Thank you. Yep. All right.
Are they going to be able to see it, or are they just going to see a blank thing?
I mean, you can see it on there if you're looking, but basically there's a screen and then there's a little corner in the corner that you can see my webcam. So if I'm in front of my computer, don't know if I can there for a minute. I might want to make use of my phone so I can actually see it.
Anybody there yet?
I don't think so. Apparently I have a subscriber. I didn't know that. Oh.
Subscribe to your channel.
I have one subscriber now.
Actually,
Yeah, no one's there yet. It's just me. That's the other thing. People will usually arrive like within 3 minutes. Maybe like a quarter of activity right now. Second. Pretty good set come. Oh, yeah. Yeah, why is it? Okay. Hello. How you doing? Hey. Welcome. How are you?
good. How are you?
Nervous, the usual. Oh, yeah. I love me some questions. Oh, yes. My parents brought all kinds of nice snacks over there, so feel free to help yourselves.
Thank you.
At long last. At long last.
Are you doing a remote session too, or no?
I have a YouTube live stream going because I have like family in Connecticut.
Tell Brendan.
I did. So.
Because I sent him the link. I mean,
oh,
not
yeah.
that it's a link, but I sent him that you were defending. But
Yeah.
I was like, I don't see it.
Yeah, I sent the link, but I don't think they wanted to put like a YouTube link on the flyer. So.
Did you send it to Brendan?
Oh, I did. Okay. I imagine there's a 50-50 shot he appears or not. So.
Earlier, he's like, go to Gonzalez defense.
Oh, he knows about it. We've been like going back and forth on text messenger. But we'll see.
He was really busy today, so I don't know.
We were supposed to like talk over the weekend on the phone, and then he just disappeared and never replied. So he is always just, how Brendan is. That's what I said. I assume 50-50 shot. We'll see. Are you taking notes?
I always take notes. Especially comes up with good questions.
Indeed.
Yeah, otherwise I'll forget. I'll have a question and then I'll go and forget it. Like I'll be like, oh, this is interesting, and then it's gone.
Yeah. My problem with note taking is always like I get too wrapped up trying to write my notes, and then I miss things people say, and then I fall behind. And every seminar, I always struggle with that.
How's your summer been, Sarah? Busy. How about you? Good. Good? Mm-hmm. Lots of data? I don't know if I'd say a lot.
good data. How about good data?
You have some good data.
Even if it's not a lot, just good.
Mhm.
Good?
You have some good data, so I'm excited.
That's what you want to hear.
That's what we want.
Mhm. Yeah, like he's coming.
Sarah, how big is your lab at this point? Because I feel like you've been like losing people. Like,
Well, seeing that I have 3 graduate students graduate this year
yeah.
and 2 last year.
Yeah, right. I mean, your lab went from one of the biggest I've seen here to I'm not sure at this point.
2 PhD students right now,
Uh-huh.
and currently 3 undergrads. Well, I have 2 undergrads this summer, but
Yeah,
one more
right,
undergrad. Well, and then I'll have 3 more in the undergrads in the fall. But I've really kind of shrunk, and I have a new master's student started.
Oh, okay. So you got a little bit of influx, a tiny bit.
I know. It
Well, the
was
master
really
students
great.
go so fast.
It's okay.
I mean, might a smaller lab like be a slight breather of management? Hello.
Yeah, I mean, 3 new grad students, 2 PhDs, and 1 this semester was a lot.
Okay.
So I think I've set myself up for that in 2 years. Again, but we'll get Jackson will graduate in the January or December, so we don't have them all the same.
I wonder if I need more seats. Probably not.
There's just chairs there if needed.
Yeah, there's a random desk here as well. I was really, this was going to be like Dave's hot seat, but I might actually put the desk there. Maybe I'll just do that now in case people want it. I don't know.
I know you
I'm
do.
thinking
You
like
need to have your
5.
committee.
Yeah, I know. That's what I'm thinking. Maybe I'll put another. Drag another desk over. Those have walls on them. All right. Oh, thank you.
I figured these chairs were better than those.
Yeah, I just realized those all have things attached to them. Hello. Awesome. Trying to find seating for everybody, or the 5, you know. The desks. I'm Dave. Thank you. No. Nice to meet you. How's my parents? Hello. Hi. Oh my goodness.
Yeah, it's been, significant.
You know the funny thing is
Yeah.
we did. I let you guys cross, but I was under conference. Yeah. Hello. And the yellow part?
I was gonna say,
No.
it was a nice ride.
Greg? Yeah. Friends host my aunt in my car. So.
I was just like, I'll just bring it back and
No, just wanted to.
Yeah.
I'm not the one.
My dad also did my car.
Happy belated birthday for you.
So I heard from Matt,
Oh,
we had a couple of texts about that, we're starting at 1.
That one's
So,
I knew that doesn't really matter. I'll start like 3.
yeah, I'll start talking at 1:05.
Yeah.
It was great.
And then you can start
To
around.
this day, I always forget the one.
I know.
Really great.
Hello, Emily.
Matt's that jumps are kind of in this.
My parents were like, that's
I
remember Joe. Seven independent. I also like 10.
Just Kelly Thomas.
Yeah.
So are you, you're all set up?
I was like, well, did I die?
Someone know.
Both of you.
I know I do.
I was like, my aunt, everybody.
After this.
And that's why I was like, I was like,
Yeah.
I mean, if there's a,
I was like, here's the group text. I've never seen this mosquito that I told Rex. What do you remember?
oh, over there.
And this was somewhere.
Oh, upstairs.
So, all right, I'm gonna start.
Oh, yeah. Oh, okay.
We saw 30 whales. Whoa.
How's it going? Thank you, Beth. Tripping on my own feet.
You had the long
I
thing
know, it
here.
was really hard to get.
This is
I didn't follow up.
I thought no one was around in the summer. Like, there's so much more people here than I thought there would be. Thank you. It's almost like teaching a class. All right, let's see.
Yeah, we'll get started in just a couple of minutes. Matt McManus is on his way from the big giant head.
Yeah, he's provost now, right? Isn't that his position?
Senior vice provost of.
Oh, okay. I can never remember the positions. He's busy. I also always forget we start at 1:10. I always just say the hour, but I guess, yeah. Almost 7 years at UNH and I still can't remember when classes start, so.
Yeah. Well, I think it's important that we change it every time.
Good point.
This week. Yeah.
Hello. Hey, Lawrence. You want to sit right up front? Oh yeah, you could grab like a little independent desk if you like. Get right up in the trees, you know.
I'm going to start talking in a couple of minutes and then it'll be right around 1:10.
I assume Anna's on her way as well. Yeah.
Here's the mix.
A lot of administration. That's
Just realized for the folks online, I'm just shifting in front of the camera back and forth constantly. It's probably slightly obnoxious.
Yeah. Who's online?
Some family in Connecticut. My I think my master's advisor from Plymouth State.
Oh.
Let's see, some of my close friends, a couple of like former graduates from here. Brendan Riley, I think is supposed to be on. I don't know if you ever met him. He was in Sarah's lab.
Oh yeah.
I know, like we were master students back at Plymouth, so we've known each other
really?
for like 11 years at this point.
Cool.
Yeah, I followed him here to his PhD.
Right. Awesome. That's cool.
Yeah, you got seven folks on there right now, so it's pretty cool.
Yeah, it's a good turnout.
Yeah, I like in the middle of summer, like close to 4th of July, I was like, I honestly don't know if anyone's even going to show up.
I know.
Yeah, 'cause you expect people to kind of take off. Yeah.
They like basically just color
Time is it? 1:04. I just realized there is no clock around here.
No clock. We're at 1:04.
That's right. Got my phone here. You barely see it, but
No, she was actually.
Yeah, and I mean, you'll you have like a 10-minute buffer if you decide to just go off.
just completely off script. Just start ranting about something completely unrelated.
I mean, did you read the Supreme Court decision?
I've read some news about this. I'm not bringing that up.
No.
Yeah.
There's all sorts of things to talk about.
Oh, there is, if you want to take that angle. State of academia funding, all kinds of stuff, Start on a high note.
Yeah. All right, everyone, we can we'll go ahead and get started. Thanks again for coming to see Chris's dissertation defense. This is something we've been working on for a number of years.
A few.
And I should say that Chris's impact at UNH, I think it really goes beyond what we're going to talk about today. I think you'll see some really interesting scientific approaches that are novel in Chris's work, and also a new software package that I think many people will use. But Chris has really had a larger impact than all of that. You've been kind of a engaged member of the grad student community for almost 7 years now.
This fall would be the start of 7 years, I think.
That's right.
So yeah, I'm running up on it.
But there ought to be a little asterisk next to that metric, because, or next to that number, because let's remember that Chris was also asked to teach as the instructor of record germs, a 4-credit course with how many students?
I had about 500 in my first semester, and then I think it was like 250 the second semester.
Right, so he was
So.
really filling in for us that whole year. That entire year you took that course on and really kind of redeveloped it into a course that, not to disparage the other version of it, but sort of had built a
It was different.
reputation.
It was different.
It was kind of a different sort of course, but Chris's version of that course really sort of changed I think the direction of it, made it maybe a little bit more worthwhile. So you did that for a year. So if you think about it, if you do the math, this is perfect timing for you, in fact.
I'll take that.
Chris has also presented at national meetings and has gotten funding to support those types of trips. We've, along the way, deployed his program in different contexts. So what you'll see today is kind of how you can start with a really basic question about molecular evolution and apply that to different types of contexts. There he is.
Hello, Matt.
I've been talking for like 15 minutes. Yeah, that's right. Anyway, so just to all of that being said, the point really is that I think Chris's impact at UNH has really kind of gone beyond some of the things that you'll see here today. But this stuff I think is really interesting as well. And you'll see how we can start with a basic question about how to deal with molecular evolution, and like questions that arise from that sort of thing, and then apply it to different contexts. So with that, take it away.
All right. Well, thank you very much for the introduction, Dave. And so, yeah, thank all of you for showing up today. The audience size is really cool. Thanks to the online people as well who are watching. I appreciate you. But yeah, so today I'll be talking about my work assessing the evolution of large gene families. I'll clarify what those are through our software program, which is phylogenetic focusing. And because I know plenty of folks, both here and elsewhere, do not work in phylogenetics, I am going to provide a little bit of a grounding framework first. But the overall outline of this talk is going to be my chapters of my dissertation. So the first one is about phylogenetic focusing itself. So I'll give you a conceptual basis of what a phylogeny or an evolutionary tree is. We'll use that context, that foundation, to understand the PhyFocus program. And then very importantly, understand the particular species data set that program is being applied to. Because the species data set actually controls the scope of evolutionary analyses here. So much of chapter 1 is all foundation that you need to understand everything else. Once we have that foundation, we'll actually go into the gene families that I've spent the past 6+ years studying. The biggest one combines 2 of the chapters of my dissertation, 2 and 3, are the chemoreceptor gene family. So I'll give you a sense first of what chemoreceptors are, the chemicals they help us sense. These are all sensory systems. And then we'll go over a subset, and by no means all, but a subset of the chemoreceptor phylogenies I've developed and worked on over the years. Once we've done that, we'll move on to my final and 4th chapter, which is about a totally different gene family called phosphodiesterases, or PDEs. It's a separate project, but the same principles. A large gene family. I'll introduce you to what they are, what they do. We'll go over the phylogenies of their evolution that I've developed. And we'll end by digging a little bit into one particular sensory PDE we're interested in, which is PDE6. It regulates our vision. Okay. So throughout all of my research, especially chapters 3 and 4, it would probably really help to keep in mind, hello, Anna, keep in mind 2 key research questions. The first one is, what are major trends or patterns that are driving the evolution of these mostly sensory gene families? And then the second one is, what are the evolutionary origins of these families? So ancient evolutionary patterns and their origins are really my focus throughout all of my work. All right, so first off, just for everyone that's not living in phylogenetics world all the time, I wanted to give some basic framework. So whenever you hear of a phylogeny or a evolutionary tree, what we're talking about is a depiction of relationships between usually, say, some species. Sorry, I used the pointer. So here, if we have, say, insects, fish, dogs, and humans, they are related in some way. These in evolutionary trees are very important. These are called nodes, these little circles. They're points of evolutionary divergence, right? You'll notice in the tree you're currently looking at, there's no sense of direction. You don't know where the evolution started and where it ended. That's because this is a unrooted tree. When you root a tree, that means you are taking a particular sequence and saying it is the earliest evolved group. So for example, if I rooted this tree saying insects are the earliest evolved group, now you have a sense of evolutionary direction in which insects diverged first, and then you have in green all these mammals, right? Fish are separate, and then dogs and humans are most closely related. So rooting is a big part of some of my trees. I'll show you later, but it gives you that sense of evolutionary direction over time. Rooting sequences are often referred to as an outgroup, so that's what insects would be here. Another major concept that I will more lightly reference throughout my talk is the idea of a monophyletic group. So this is, for example, in green. Monophyletic group just means all of the things that have diverged from a singular node. So if I look at this node right here, I would say all of these in green are vertebrates, animals with an internal skeleton, right? Fish, dogs, and humans. Very commonly, just to get away from terminology, I'll refer to this as groups throughout my talk. So when you hear me saying groups, I'm usually referring to a particular clade or subset of a tree. Now, this stuff is all just based on species, but my entire work is actually on assessing the evolution of genes, right? Fortunately, species trees and gene trees all employ the same conceptual framework. One big caveat, though, is that gene trees, understanding the evolution of different genes, requires sequence data, right? Sequence data is a huge component of gene trees, whether it's DNA sequences or protein sequences. Everything I show you today will be based on protein sequence data. All right. So on that point about sequence data, in the past 10 years, this is just from a government data set called GenBank. So in the past 10 years, from 2016, you can see the billions of sequence records available in that data set has skyrocketed. And for those interested, like I am, in building evolutionary gene trees, this is actually really valuable, right? More sequence data you have from the more variety of species, the better and more deeper your evolutionary analyses can potentially become. Also important, I'll just note, while a lot of this growth in sequence data is from viruses and bacteria, the number of animal genomes is the whole set. Number of animal genomes available has exploded as well, and not just from our classic models in science of things like mice, humans, or like fruit flies. However, there is one caveat for making gene trees on sequence data, which is you cannot just take, unfortunately, you cannot just take all of the sequence data out there and throw it into a tree to understand how all of like DNA evolved. There's two major aspects of that. Too much highly divergent sequence data can actually severely hamper or limit the quality of your final gene tree. It also severely increases the computational time to make the gene tree. I will show you an example of one gene tree that took over a month to make, running constantly on a server because of how large it is. And that's approaching kind of the limits of what you can do. The point being, you have to limit the sequence data you take in. So you need some way of selecting that. Now, in sort of a very broad brush classic approach to selecting sequence data, I'm just going to outline here a very basic approach. There's a lot of iterations to it. But the basic idea is, let's say, for example, we have taste-1 receptors, a certain type of chemoreceptor we'll talk more about. If that's our target, the thing we want to study the evolution of, super common approach is you'll take taste-1 receptor sequence and blast it using the program BLAST. It's a search tool. Blast it against, say, that GenBank, huge data set of sequences, and try to find any that are similar, right? So just say, find any sequences similar to our T1Rs. Generally, there is some form of a subjective criteria to limit how many sequences you get back, right? That's the way you constrain some of that available sequence data. Subjective limits most commonly include the number of hits. So you might say with blasting, you might say only find the top 50 sequences that are most similar to our taste1 receptors. A more common, I think, metric people rely on is a e-value, like a statistical test for similarity. So you might set a particular e-value, and any sequence that doesn't match that e-value just gets tossed aside. Many researchers, if you look at the literature, will often manually add or remove sequences as well, just based on their own prior knowledge or the research literature. Now, I do want to stress that once you've done that, the process, painting broadly here, becomes pretty simple. You then align the sequences you have, you apply an evolutionary model to them, and that's how you ultimately construct a gene tree. Okay, that whole process can be quite complicated, but simply put, that's how it works. Now, I do want to stress that this classic approach is actually quite effective. Highly similar sequences usually do have some form of evolutionary relationship, not always. So searching for highly similar sequences and using these subjective criteria can produce and does produce informative evolutionary trees. The problem is that depending on the specific, especially if you're trying to study highly divergent sequences, this process can cut those out of the picture, right? So the classic approach will get you evolutionary trees that depict real relationships, but there's a real chance you will be excluding the full picture, right? You'll be missing data. And so we wanted to try to address that, to find another approach that can try to, especially for studying deep origins or evolutionary history, try to understand all of the possible relationships that are out there in the complete data sets for sequences. So this is where we get to phylogenetic focusing and what it's trying to do. So conceptually, phylogenetic focusing is based on this idea of a phylogeny-first approach to sequence selection. This was proposed by other folks, but phylogeny first, the name pretty much tells you the approach. The idea is out of that mass of starting sequence data, take those that are pulled in, construct a starting gene tree, and then just look at your target in the tree and take those sequences that are related to each other. Basically, use evolutionary models to select the sequences for you rather than subjective criteria, right? And so this was originally proposed for other projects called like ortholog group assignment. So the idea of phylogenetic focusing is take that phylogeny first approach and apply it to gene trees, okay? So that's what the software does fundamentally. I'm going to step you briefly through the 6 major steps of the software, just so you have a sense of what's going on when I made my trees. We're not going super deep on this, but step 1 is very similar to the classic approach. You use BLAST with your target, say our taste1 receptors, and try to find as many similar sequences as possible. Note here, though, that the BLAST is highly permissive. So we don't try to use those subjective criteria of like stringent hit limits or a stringent e-value. We, in fact, make them as lenient as we possibly can. For example, the hit limit is 5,000 by default in the program. So the goal is basically give me anything that is remotely similar to, for example, taste1 receptors. Once you have done that for each species you are assessing in your data set, we construct individual gene trees, so 1 per species. So it keeps the gene trees relatively small, so you're not making a monster tree all at once. And this is where the focusing element starts to come in. So in phylogenetic focusing, you first have to specify a rooting or outgroup sequence, like I mentioned earlier. So you need that evolutionary direction in the tree. So like here's an outgroup, you have your target. And then we have one unique thing that we actually bounced around a bit figuring out what to name it, but we call it an anchor. So you have to provide this at the outset, but an anchor is a known relative of your target. So you don't maybe know exactly how it's related, but you know it's related. So for each species, you make these initial gene trees. You can see outgroup, anchor, and target. And then in step 3, you actually focus. Focusing means you look for the most recent common ancestor of the anchor and target. So if we look over here, you can see this node that the arrow is pointing to is the most recent common ancestor. You take those sequences, you cut them out, and you throw away everything else. So this is phylogeny first, right? Selecting a subset of sequences based on evolutionary models and then discarding the rest. Once you've done this step, it's basically the rest of this is the same, essentially the same pipeline as the classic approach. You take all of those individual gene trees, you mash them together to make one grand alignment, and then you apply a model to produce your final evolutionary tree. Okay. So that's just the overview. Again, I could do a whole talk on just that, but we're not going to do that. That's phylogenetic focusing. I will also just add that in chapter 1, I did a bunch of tests manipulating variables in the various steps and comparing those outputs to the classic approach, just to get a sense of how the program is performing. I could show you some of that afterward if anyone's interested. But the short story is that I would argue phylogenetic focusing provides viable trade-offs both in computational time and tree quality. It does cost more time depending on how you do it, and it can hinder a little bit tree quality, again, depending on how you do it. But I would argue it's worthwhile because it allows you to have that phylogeny first, very inclusive approach and assess far more species than most classic approaches normally do. But like I said, I talk more about that later. I will also show you a QR code to my GitHub if anyone wants to look at the user manual for PhyFocus, which I have on there. But anyway, let's move on. So whenever you're doing any sort of evolutionary phylogenetic analysis, you need to have a data set to work with. So for everything you see today, this is going to be the data set. It's 45 species, 43 of them are animals. 2 of them are just non-animal single-celled eukaryotes called choanoflagellates. And remember, our main research goal here is to understand evolutionary patterns and origins. So because of that, we really tried to stress in this data set a lot of ancient animal lineages. There's 3 major groups I would ask you to try to remember, and I will keep showing this throughout the talk. The first one in purple is non-bilaterians. These are things like sponges, jellyfish, ctenophores, very ancient lineages of animals. In green, we have the protostomes. These are ones a lot of people don't like. Things like insects, arthropods, crustaceans, octopi, for example, mollusks. And then finally, our biggest group in various colors is the deuterostomes. We've got 19 species of those. And these include things like the vertebrates, right? Animals with internal skeletons, including ourselves. For these, just so you know, I tried to color code them in an intuitive way. So aquatic vertebrates are in different shades of blue. And then once we get to the terrestrial ones, I used warmer colors. So like birds and chickens and reptiles are in orange, and mammals, including ourselves, are in red. So we'll be looking a lot for the presence or absence of these 3 groups in different clades of the gene trees. All right. So I know that might have been a lot of foundational stuff, but I think it will really help understand the trees we will now be looking at. So I'm going to begin by getting into what is by far my biggest chapter, chemoreceptor gene family evolution. And again, looking at major trends and patterns and origins. So first off, for everyone who's not aware, what are chemoreceptors? So these are membrane-bound proteins, usually in nerve cells, neural cells, and they are key for detecting chemicals in our environment. The ones we most commonly would think of is our senses of smell and taste. Those exist purely because we have chemoreceptors. And that can take really different manifestations based on the species. Like for vertebrates like ourselves, we think of mouth and nose. But like for an octopus, for example, they can use chemoreceptors to smell and taste using the tentacles and suckers on their arms, right? So a lot of different manifestations of how this sense is expressed. But chemoreceptors do more than just this. They are also important for social communication in many animals. Think of, for example, the social insects, right? Ants, bees, that kind of thing. Many animals as well. So pheromones are all detected through chemoreceptors. Especially in like many mammal species, maybe not so much humans, but many mammal species, this is a key part of like sexual reproduction and reproductive behavior amongst other social dynamics. And finally, I'll just point out that for a number of species that have highly reduced senses, say highly reduced vision, think like blind organisms at the bottom of the ocean or in deep, dark caves, or things like, for example, our hagfish and hydra, chemosensation of the environment may be one of their main and primary ways of interacting with the world around them, right? They lack, say, conventional eyes. All right. So given that chemoreceptors have played such a highly diverse and important role in animal evolution, it's probably not too surprising that these genes are incredibly broad in terms of how you categorize them. There's 10 major gene families I worked on in my dissertation. There are others. I will say this is not an exhaustive list, but these 10 are sort of the big ones. And they break up into two major gene groups. 7 of them belong to G protein coupled receptors, or GPCRs for short. Those are most commonly known in deuterostome animals, such as vertebrates like ourselves. The 3 other major gene families are ionotropic. They are based on ion channel signaling. And those are most commonly identified in protostomes, especially insects. Okay, so we'll be taking a look at both of these major groups. All right, so first off, kind of start with the biggest one, GPCRs. GPCR superfamily is the largest group of genes known to exist in animals. It's insanely large. And it consists of several major groups. For our purposes, fortunately, we don't need to look at all of them. Chemoreceptors have only been identified in the class C glutamate group and in the class A rhodopsin group. So those are the 2 we're going to zoom in on. We'll start with the class C or glutamate GPCR group. The class C GPCRs have 2 chemoreceptors in them. They have the taste-1 receptors. These are required for our senses of sweet and meaty flavors. That's how you detect that stuff. You're using taste-1 receptors. And then there's vomeronasal 2 receptors. Humans, as far as we know, don't have functional V2Rs, but they're used for pheromone detection. All right. Now, you might remember in the 5-focus approach, ideally, if we're going to do the full focusing, we need to have a rooting group, right? Something of an out group, and we need to have an anchor, right? Something to focus in on. Our targets, of course, would be the chemoreceptors. So there's 2 neural signaling genes in this family called GABA receptors. They're widely recognized as the earliest evolved group, so I used those as the root. And then I anchored with a group called the metabotropic glutamate receptors. They're just a large clade, generally thought to be outside the chemosensory genes. So when you do all this and you run 5-focus, here is the phylogeny you end up with. Okay. And of course, I included the colors over there so you could still kind of track what's going on. But a couple of things. All my trees are made with the maximum likelihood methodology for making phylogenies. And this particular class C gene tree is built off of about 2,700 sequences. So each tip represents a single sequence, a protein sequence. I will also note that a number of these nodes have little circular markings on them. Those are nodes of interest that I was writing up in my results and discussion. And they are color coded based on statistical tests that are run on every single node in a phylogenetic tree. These tests just simply determine whether the branches off that node were placed in a statistically significant way or if they were not. So black circles indicate really strong support. Gray is in the middle. White is weak support. So white nodes, for example, if I made this tree again, the white nodes could potentially jump to different locations. Some of these I have actually observed up here or over here as an example. All right. So let's take a closer look. One thing I want to do is point out our anchor, the metabotropic glutamate receptors, because these really nicely illustrate what an ancient gene family will look like in this context. So I should use the pointer. If we look here at the beginning, you'll notice there's all these purple sequences. Most of these are cnidarians, so they fall into the non-bilaterian group at the very base of the animal tree of life. And as you move down this part of the phylogeny, you'll notice there's a sliver of green here. That's our protostomes. And then you see the range of colors that represent our deuterostomes. So what you're seeing is the entire animal tree of life recreated here. That indicates that the metabotropic glutamate receptors originally evolved somewhere at the very origin of animals, because all animals are present there. Furthermore, if you look a little further, you'll notice some of the colors from the protostomes in green and then the range in deuterostomes repeat over and over again, right? Especially with the vertebrates. You see these like red and orange colors repeating. Those are gene duplication events. So that means sometime in the evolution of vertebrates, there was gene duplications where metabotropic glutamate receptors duplicated in the, like say, chromosomes over and over again. So that's what we're seeing there. So let's compare this ancient gene family to one of our first chemoreceptors. So if we look further up, here are the V2Rs, right? The vomeronasal nasal 2 receptors. One immediate contrast, I'm sure you can notice, is that the duplications are far more intense, right? Light blue here is mostly the fish species I had in the data set. And you can see fish alone have a huge swath of duplications. This aqua is amphibians, then we have birds and reptiles, and finally mammals, right? So what this is indicating is that over the course of evolution, even down to particular subgroups, like say just mammals or just amphibians, there is a strong conservation of any time something is duplicated in this field, it isn't lost, right? Those genes do not just simply become lost or become pseudogenes. They seem to take on presumably new functions and be conserved, right? Now, in terms of their origin too, I also want to point out, notice there are no purple tips here, and there are also none of the main green tips here. So there are no non-bilaterians and there are no protostomes. This seems to suggest that the V2Rs originated in the evolution of some part of vertebrates, right? Now, for a long time, if you look at the literature on this, it was thought that they originally evolved in the what are called gnathostomes, the jawed vertebrates. If we look at the species list here, gnathostomes are everything after the hagfish and the lampreys. So basically fish, sharks, and on, right? So it's a subgroup of vertebrates. However, when we ran this tree, we noticed this other really dark blue clade right here. And this is a clade entirely of hagfish sequences. So they clearly duplicated a bunch, and you can notice it branches off of a node that connects to the rest of the V2Rs, and this node is very highly supported. This strongly suggests that, even though it wasn't reported in the past, hagfish seem to have some gene sequences that are related to, or are in fact are V2R genes, right? So if that is the case, this would definitely suggest that the origins of V2Rs is actually at the beginning of the vertebrate clade. So that would be like 50 to 100 million years earlier than originally thought. I will note this has been supported actually because last year, another research group actually published this data. So I got a little scooped on that. But they found the exact same thing. They did a phylogeny like this, and they pointed out in their paper, hey, look at this massive hagfish clade right next to the V2R gene family. So this is becoming the established, I think, expectation that V2Rs evolved originally at the root of vertebrate species. So that's just the V2Rs. We do have one other chemoreceptor here, right? Our taste1 receptors. So if we look here, you can see them, the T1Rs clustered a little bit in between some other gene families I won't talk about. So what I'm going to do is pull out that subclade so we look at it more closely. So this is the same part of that tree just blown up so we can see it. And the taste1 receptors actually have 3 subfamilies. This is well established in the literature. It's taste1, R1, R2, and R3. And these genes work together to enable our sense of taste, basically. I forget exactly how it is, but I think it's R1 and R3 work together for a meaty taste, and R1 and R2 work together for a sweet taste. I think I have that right. But the point is they work together. And so we have the expected subgene family clades here. And if you look at the colors in our R1, R2, and R3 groups, you'll notice there are none of the dark blues, right? Those hagfish or lampreys. So this suggests that the T1Rs actually evolved after the V2Rs, right? They originated with the jawed vertebrates, the gnathostomes. So a little bit later in our tree of life down here. Particularly interesting, or at least I found it quite intriguing, is that there's some of the clades in my tree that quite frankly don't make entire sense, right? So if we're looking at the groups, you'll notice, for example, for T1R2, there's a group here spanning a wide array of species of vertebrates, but then what's called sister to that, right next to it, there's another lone group of, these are coelacanth fish species that are just by themselves. That seems a little odd. Same thing down here in the R3, there's a large taxa vertebrate spanning clade, and then there's a sister clade just kind of sticking off to it with a scant few species in it. Most bizarre, though, to me, was this group right here in the middle. Notice it falls entirely outside of R1, R2, or R3. It's like it doesn't belong to any of them, which to me didn't make sense. Originally, I thought maybe these were just highly divergent sequences that were kind of as a fluke, just not sorting properly in the gene tree. That can happen. But then when looking at the literature, I found a very interesting paper from 2023. And they made the bold assessment. They were assessing the evolution of taste1 receptors as well. They made the bold assessment that there are, in fact, not 3, but 8 taste1 receptor subfamilies. And this is a modified version. I flipped it around a little bit. But this is basically the phylogeny they proposed. And what's really kind of cool is if you take the phylogeny of my own that I just showed you and apply their labeling, you can actually see almost all those groups. 7 of the 8 are readily identifiable using my tree. So again, like some of those mysterious ones that I didn't know what they were, they, according to this 2023 paper, would be R6 and R7. And then we have sister to R2, we have R5, and then R4 to R3. Now, I will say I'm not entirely convinced just based on this that these are completely novel, like 5 never before known subfamilies of taste1 receptors, in part because there's not many species involved in some of these identifications. So I would argue this is a good spot for, say, future work to make a, say, vertebrate specific data set and try to really flesh out how many species actually have these clades and are they well supported and conserved. Furthermore, I think the best approach ultimately would be to try to do some functional work on what some of these supposedly new subfamilies are doing, right? Because taste1 receptor biological function has been studied for decades. And if there are 5 unknown gene families in this group, I would wonder, like, functionally, what is that doing for our taste modalities? Do we have like tastes that we've somehow never identified, or are they overlapping in function with the R1, R2, R3 schematic? Right now, I have no idea, but I think it'd be very interesting to delve further into that. All right. So those are the major components of the Class C GPCRs with those 2 chemoreceptors. In the interest of time, of course, I'm not going to dive into every single subsequent gene family of the chemoreceptors. I would be here for probably 2 more hours. But I do want to give you some overview because some of it's really cool and I at least would like to share it. So if we take a look at the next group, the Class A GPCRs, the Class C was a wonderful testing ground for Phyfocus. Class A was, relatively speaking, a nightmare. And that's because the Class A GPCRs are massive and incredibly complicated. So based on this paper, Fredrickson, which I often used as a reference, 2003, phylogeny of Class A, there's 4 major subgroups, alpha, beta, gamma, and delta. 3 of them are considered to have chemoreceptors, as you can see here, alpha, gamma, and delta. So initially, what I tried to do is I tried to apply Phyfocus to each of these subgroups independently. I'd look at alpha, I would choose an anchor and a root. Of course, we had our chemosensory targets, and I would try to Phyfocus. What would happen is the tree wound up a bit of a disaster because many of these sequences actually seem to have related elements to each other. And say in my alpha Phyfocus run, I would start getting gamma and delta sequences. In my delta run, I would have alpha, beta, and gamma. Basically, I would keep recovering a whole slew of others that did not seem to sort as monophyletic groups as we might expect looking at this. So given the struggles of proper rooting and anchoring, I eventually decided to just take everything and try to make one massive Class A tree. This is where the part I was mentioning about the importance of sequence selection matters. This was very difficult to do. So when you try to do that, this is actually what I wound up with. This took at least 40 to 50 days to run using, I think, 128 gigabytes of RAM on a server. And this entails about 16,000 tips. So about 16,000 protein sequences are entailed in all of this. I know you cannot read the black lettering. That's expected. But basically, this is just me trying to denote all the different Class A receptors that are identified in various clades. The short story is that most of them are receptors for either neurotransmitters or hormones. So you have everything from like steroid receptors in here. There are some that are a bit different, like for example, opsins for vision. And of course, buried in all of this, we have our chemoreceptor groups that we were initially interested in. So if you kind of ignore everything else, basically I ended up pulling out 4 subtrees containing the various chemoreceptors. These two, the V1R and T2R, different from the ones we just saw earlier. These actually sorted together in one tree. So I wound up with 4 trees for 5 chemoreceptors. What I'm going to do now is just give you a very simplistic overview summary of what I found in terms of trends. I'll talk about origins a little bit in a moment. But for example, here is the olfactory receptor subclade. So olfactory receptors are the ones responsible for smell. ORs, the OR family is considered the largest gene family known in animals. Mice and rats, for example, commonly have anywhere from 600 to 800 OR genes, right? So if you can't tell, mice and rats would be the red. Gene duplication is the story here, by far extensively beyond anything else. That really is what drives their evolution. And if we're talking about ORs as an example, olfactory receptors, you'll notice it's not just evenly across the board of all animals. There is some gene duplication, for example, in fish. We got the blues down here. But you'll notice the vast majority of them are in the land animals, right? Orange being birds and reptiles, reds being mammals, okay? So it seems like land vertebrates really took advantage of every gene duplication event to develop more olfactory receptors. Given the like millions of different chemicals you can smell in your environment, that does make some inherent sense, I would argue. By contrast, let's take a look at a different class A family. So these are the trace amine receptors. These are chemoreceptors that detect proteins in your environment. So they're specific to certain peptides. And here, duplication is once again the main story, but you'll notice the species involved are quite different. So we have all vertebrates. Here's the hagfish at the very beginning, and then we have the full color spectrum. But I'm sure you can see pretty clearly the blues are far more predominant here. So trace amine receptors, much more of a thing in fish than say in mammals or reptiles. And if you think about their environment, right, fish are aquatic. Proteins that are dissolved in the water are probably a really important thing for them to detect, right? So there's a much more stronger evolutionary pressure for them to develop any duplicated genes in this family as new chemoreceptors. All right. Like I said, I was doing a very brief overview there. If you want to see more of these trees, I can certainly show them to you later. But that's all I'll say about them for now. I do want to wrap up the chemoreceptor portion by talking about the one group I often feels a little neglected, which is the chemoreceptors of protostomes. So think things like insects and arthropods, my personal favorite group. 3 good families here: ionotropic glutamate receptor, or not glutamate, ionotropic gustatory receptors, ionotropic olfactory receptors. Do not confuse these with these, they are different. And then, rather confusingly named, there's also the ionotropic receptors. IRs, I'll just say. For our purposes here today, I'm just going to show you the IGRs and IORs. They group together in one tree. And that one tree looks exactly like this. So one thing you can notice right off the bat is the number of sequences involved here is way less, right? Compared to the like GPCR trees I just showed you. You can actually see the names of everything here. They're not all mashed together. So far fewer sequences involved. The IORs and IGRs pretty much only found in some protostomes. Even then, not all of them. It's mostly insects, honestly. But a couple of interesting points here, if we take a slightly closer look, is while they are mostly found in arthropods and other similar protostome species, right at the very beginning of the tree, I was somewhat surprised to find a bit of diversity, right? So we have a couple of jellyfish sequences. So that's our non-bilaterians. And then we also have a couple of sea urchin sequences. Sea urchins are one of the earlier groups of deuterostomes, right? So in terms of the origin of this whole gene family, this actually suggests that despite the fact most of the genes are in arthropods, the origins are actually way back, perhaps with the origins of animals. the fact that other animals like vertebrates don't have them seems to imply that a lot of species lost this gene family over time. So it appears to have originated at the root of animal evolution. Deuterostomes, except for the very early ones, lost these completely, and only select protostomes, mostly again insect related, actually diversified and evolved more of them. So that's a major trend in terms of origins. I will note one other point I didn't mention. The ionotropic gustatory receptors are largely recognized in the literature as being like the originator clade. Within them, sort of diverging from them, are the ionotropic olfactory receptors. So this clade in gray was once a gustatory receptor. They diverged at some point in ancient history and actually changed and mutated so much they became their own gene family. Now, these ones carry the trends I just mentioned sort of to the hilt. So ionotropic olfactory receptors, again, used for smell, like the name suggests, only identified in insects. Every single tip I have here is from the fruit fly. Nothing else in my data set matched to it. And when you look at the research literature on this topic, it's basically the same story. Even if you include a bunch of other protostome species, only insects come back having these. So this is a very specific set of gene duplications for one particular taxa of organisms. And again, duplication, at least in insects here, is still a very big component of their evolution. All right. So chemoreceptors, big final summary here. As I've been saying quite a bit, group or species specific duplication clearly is the main driver of their evolution, right? Every time genes duplicate, there's a clear like selective pressure, probably on the basis of say ecological or various say dietary niche adaptation to take those gene duplications and specialize them for some functional purpose, right? So duplication, very big component. If we consider, say, the protostome deuterostome split a little bit, for protostomes, the history seems a bit more limited. Obviously, certain groups like the IORs are hyper-specific to a small group like insects. Well, insects are not small, but for our purposes, a single group. While some of the others, like the ones I didn't talk about, IRs, are found throughout protostomes. But with deuterostomes, right, so if we shift down here, the story is a little bit messier, as you can tell from all my arrows and annotations. But what's interesting is that most of the origins I identified based on my phylogeny seem to cluster around 2 points, which are these red lines on the tree you're seeing. And they're labeled 1R and 2R. For folks that don't know, this is the 2R hypothesis of vertebrate evolution. It's the idea that in the very beginnings of vertebrate evolution, there was a whole genome duplication event. That is 1R. So the entire genome of the vertebrates completely copied itself. Then after the origin of vertebrates, but at the origin of gnathostomes, the jawed vertebrates, there was another whole genome duplication event. That's the second R, right? And you can see that based on my trees, the origins of most chemoreceptor families of the GPCRs are all clustered around these 2 points. Now, that doesn't mean definitively that they base their functional origin in whole genome duplication, but it is a correlation, right? It seems very possible that that played a significant role in the evolution of these gene families. All right. So let's take a break from chemoreceptors, a permanent break, I suppose, and we will move on to chapter 4. Right, totally different gene family here. These are the phosphodiesterases or PDEs. So this is a project that Dave and I worked specifically with Rick Cody because he focuses on like vision evolution and signaling of the phototransduction pathway. So that was my entry point into this. And like before, I'm just going to give you a little bit of a basic overview before we look at the trees. Okay. So first off, PDEs are an incredibly diverse group of genes. They're not as broad as a lot of the chemoreceptors, but they do many different things. So they have been identified, or PDE-like genes have been identified in bacteria, archaea, and eukaryotes. So they seem to span in some form the entire domains of life as we know it. Within animals, there are 11 PDE families that are known. Those 11 families contain 21 distinct genes. And as you can see from this table here, the families have biological functions that spread pretty much almost to like every major system of your body, right? They're very diverse. One of the things that did draw our interest, especially given our interest in like sensory systems, is PDE6. It is the only one of the PDEs that we know of that has a sensory role. It is involved in your retina. It plays a key role in vision, right? If you don't have PDE6 functioning properly, you go blind. Oh, I forgot to mention all PDEs, one thing they share in common is they regulate cyclic nucleotides. So they're a big part of cell signaling regulators. That's why they have such diverse functions. So when I tried Phyfocus on this, here is the tree I got. Again, pretty busy, a lot going on. But for one thing, I can't just say in the research literature, when I started on this, there was only, from what I could find, only like I think it was two published papers that had a PDE family history, phylogeny, and all of them were just based on human and nothing else. So very limited. So I was curious to see if the full evolutionary history with a Phyfocus approach would match the literature. And as it turns out, it pretty much does almost exactly. So major trends that I'll just summarize is PDE 1, 4, and 3 tend to cluster together, often as a single clade. That's a little harder to see in this. But if you look at like the unrooted version of this tree, it's pretty obvious. PDE 7 and 8 often appear closely together, but their placement can vary a little bit. Some of the node support is actually weak at that point. PDE 9 is consistently an outgroup to what are called the GAF PDEs. GAF just refers to a portion of the protein, a domain that helps them bind cyclic nucleotides, if I'm not mistaken. So GAF PDEs are known to be a subfamily. And of course, here in my phylogeny, they all group together with PDE 9 as the outset. So despite including 45 species here, pretty much matches the human-based evolutionary trees I've seen in the literature. So good support for that. A couple other points when we're thinking of, again, broad patterns and origins is for the first time in my data set, some of those non-animal single-celled eukaryotes at the very top actually appeared, okay? Not in all of them. It's actually, I think, only like 4 or 5 of them, but several times they appear. So that does suggest that even the animal PDE gene family or gene families actually do have origins much earlier than the evolution of animals themselves, right? They appear to be found in non-animal single-celled organisms, at least to some degree. So that was a point. Another one, and this is again sort of a similar theme, is gene duplication seems to have a major impact on PDE evolution. However, unlike with the chemoreceptors, where in a, say, chemoreceptor clade, you would see it kind of across all the species included. Here, it only seems to really impact vertebrates, right? So many of these like clusters are usually like isoforms, just different chopped up versions of the same gene. That's why you're seeing like many of these. But when you look at an entire replicated group, you see, for example, PDE1C, B, and then A. So in this case, duplications that resulted in 3 vertebrate-specific PDE1 genes. And whenever you look elsewhere in the tree, you basically keep finding these same patterns. PDE4 has 4 distinct vertebrate-specific gene duplications, right? And ultimately, if we go to our single sensory PDE, PDE6, again, vertebrate-specific duplications, right? I will say, as an aside, I'll kind of touch on PDE6 slightly more. We're about to wrap up. But you'll notice here, actually, that the branch length for PDE6 is also really long. Branch length indicates how much a sequence has changed over the course of evolution. So from here to here, if the branch length is really long, that means these sequences are very different to the ones that came before. So clearly, especially with vision PDE6, something in vertebrates really changed drastically, right? Excuse me. All right. So, oh, hey, what's happening? All right. So just to wrap this up then, animal PDEs 1 through 11, origins seem to stem quite frankly, off my species group here, off before the evolution of animals. When we zoom in to look at these habitually duplicating vertebrate PDE genes, once again, a really tight correlation with that 1R and 2R grouping, right? In fact, every single one I found that had multiple vertebrate PDEs all grouped exactly here. And I found some supportive literature from the PDE6 studying its chromosomal placement, like synteny analyses. And those papers were suggesting that PDE6 was actually based on, I think it was 2 or 3, I think it was 2 arms of human chromosomes. So indicating again that some form of like genome duplication had allowed these different groups to appear. Excuse me. All right. Last thing I'm going to note is about that long branch length. That is particularly distinct because while I've been saying PDE6 plays a role in vision, it only plays a role in vision for vertebrates. So PDE6 in non-vertebrates, like for example, protostomes, is not known to have any role in vision, right? So something about the dramatic change in the sequence here presumably had something to do with evolving a vision-based function. And that is something that we are trying to address. And actually, I'm sort of in the process of wrapping up our analysis on that, which is why we're not talking about it, besides the fact I'm out of time. But that is sort of a very near future directions. So speaking of, our basic idea here is to use protostome PDE6, non-vision, and vertebrate PDE6 that are involved in vision, compare them and try to figure out what sequences or what mutations occurred in the vision version, right? So we're using a program called CAST tools to do that, to identify sites that have changed only in the vertebrate PDE6. And then we're mapping those using Chimera X onto a model, 3D model of PDE6. A very preliminary finding that Dave has very much helped me clarify, so thank you, is that it seems the sites that are different in the vertebrates appear to be potentially correlated with the binding sites of a vertebrate-specific PDE6 inhibitor. So if that turns out to be true, that strongly suggests that the evolution of vision in vertebrates through PDE6 is correlated with the evolution of this vertebrate-specific inhibitor. So hopefully we'll kind of wrap that up in the next couple of weeks. More broadly, last thing I'll say about future directions is that everything I did in my dissertation was very broad focus, right? That massive animal 45 species data set, looking back at origins and specific aspects of evolutionary patterning and time over time. But a five focus approach, or even just a gene tree approach in general, can really help you answer questions about almost any genes you are interested in. So if we think about, for example, the strangeness of taste1 receptors in vertebrates, you could just change the data set, make a 50 species vertebrate data set, and then try to do a five focus, sorting those vertebrates by, for example, dietary preferences. Then you could look and see which animals are appearing in which clades. How does that correlate to their dietary preferences? What does that tell you about the evolution of, say, adaptations to different diets or environments? There's many examples you could think of by making your data set based not on ancient history, but rather some particular aspect of an environmental niche or adaptation you are interested in. So that's my plug for researchers in the room to consider gene trees in the future. Anyway, that's the end of my talk. I do want to give acknowledgements, and I know the slide's a little packed, but there's a lot of people to thank. Of course, I want to thank the committee members that are in the room. All of you have been obviously a great guy, especially my primary advisor, Dr. Pachetsky, who has helped me out with a bunch of things and also been super patient in a bunch of ways while I'm bouncing around between teaching and research. I also want to throw out thanks to Dr. Chen and Fudge, who were on my committee originally when I was focusing more on hagfish physiology. So they left my committee when I changed my focus, but they really helped in the beginning with advice and guidance. UNH COLSA, and of course the MCBS department for various forms of funding, opportunities, as Dr. Pachetsky mentioned, to teach. Those meant a lot to me. I really like teaching, so that was huge. I really appreciate it. For coding, mentorship, Drs. Tony Westbrook and Easton White were huge. When I came to UNH, I knew LS was a command, and I also knew the command line and terminal you should probably not touch. Being able to make PhyFocus being a software functioning program on GitHub is purely because these two helped me out. Tony Westbrook in particular, because I failed his course the first time I took it, he really gave me a lot of mentorship in that. So I appreciate it. Thanks also to past members of the Pachetsky Lab. So Hannah Pear, or Pare, I always mess up her last name, sorry. Hannah contributed one of my species data sets, which I need to talk to you about how to cite that. Sydney Birch, who I think is here online, really great source of support. When I went to the SICB conference off in Texas, she was the one friendly face I knew who kept dragging me around to places. So I'm kind of a shut-in, so that was really great for me. And then I want to give thanks to Curtis Provencher as well, who was one of Dave's past master students. I never met him, but his master's thesis was the conceptual original start of PhyFocus. So he deserves credit and recognition. Finally, I want to thank family and friends. So my parents in the back, Kim and Kevin. There's a lot to say there. I mean, basically, like as a teenager, I never thought I would go to college. I didn't think it was something I could do personally. They kind of sacrificed a lot and made a great deal of effort to make it like financially possible for me to go to college at all. And ever since then, in my academic meanderings, they have always done their best to support and encourage me in whatever I choose to do. So you've been wonderful parents and really great friends, and I'm really appreciative of everything you've done. I also want to thank some really close friends that have been in my life for quite a while, Taylor Hornig and Dr. Brendan Riley. A bunch of you here know him. I knew Brendan like back in our master's time. So both these people I've known for nearly or even more than 10 years. And they've endured unending hours of my rantings and ramblings. They've been a huge, you know that, they've been a huge source of support and really like bring a lot to my life. And I'm really grateful for both of them. And then finally, all sorts of people, including many of you here that I know, extended family, friends, and past teachers. You know, you all contribute to my life in many ways, and I'm grateful for you. So with that, I will share some of my last favorite seminar jokes. So for those of you who've seen these in seminar for the past 7 years, here's your last look. Not a joke is the QR code for my PhyFocus program, if you would like to look. And I will take any questions you have. Folks online, if you have questions, text me or try to communicate with me some way, and I will get back to you. Thank you, everyone. Yes. Thank you. Oh, the gustatory receptors? Yeah.
And then a couple the non-experience and sea urchins, whichever ones those were.
Let's see. Yep.
You mentioned that possibly vertebrates lost this ability, and forgive me, this is a naive question, but
no.
Is there any chance that those two sort of evolved them independently, and they were just so similar in sequence that they map this way, or like how do you know?
Oh, you mean so like the IGRs and IORs? Like how do we know they're one group? Yeah.
Back here. But like, how do we know that those are coming from the same origin of beginning of animals versus them like evolving them? Getting word like Dr. Chensky, but
Convergent.
Oh, like so like convergent evolution. Okay, thank you, Dave. So yeah, that's a great question. Some of the other trees actually, I won't go off your question. I'll just say some of the other trees actually did have, like class Cs, actually had a very distinct example of convergent evolution that I haven't mentioned. But yeah, in this case, that is a little hard to say for sure. It can't be some instance of convergence because I don't know this, but I suspect we probably don't really know like what these jellyfish or sea urchin sequences are specifically doing. I doubt they've been functionally assessed. I mean, function can diverge as well. So offhand, yeah, I can't say for sure that this isn't potentially an artifact. That is possible, in part because also we don't know entirely how to root this tree. As it appears, it looks like it's rooted as a direction. I allowed it to stay like this because we do know based on a lot of previous literature that these IORs are a subgroup of the IGRs. So this direction follows that, expectation. But yeah, where exactly these would be placed, I don't really know exactly. So it's a great question. The answer is we're not certain. This is all, I should say, all gene trees, all phylogenetic trees are hypotheses of evolution. There's always a possibility that they're not depicting everything, exactly right or correctly. So this is a working hypothesis, I would say that.
Good answer. I would also add, could you go to the PDE6 tree?
Sure.
These analyses, like Chris says, they allow you to formulate hypotheses on convergence. And one thing we noticed when we looked at the PDE6 phylogeny, I'm not sure if it's this. Oh, yes. So you can see, will you point out the octopus sequences that are?
I'll go to the next one, which I think blows it up a little more. This is where animations are annoying.
Yeah.
There we go.
Yeah.
So yeah, octopus is right here.
There's a clade of octopus sequences that are out of place. And that's not really where they ought to go if this tree is following the phylogeny. So there's
Right.
other types of molecular evolution going on.
So it's going to this node right here. So yeah, octopus is sorting away from the other protostomes and it's sorting with the vertebrates,
So
which
we
clearly
might-
is not normal.
We could hypothesize that in the octopus, there is PDE6, still a PDE6, but it's evolving convergently to have sequence similarity to
Right.
individuals. And maybe it's an independent origin of PDE6 based vision in invertebrates,
Right.
which would be kind of cool. Or maybe it's not that at all, but that would be the kind of inkling that could get us to test that.
And just building off that real fast, I know I'm really stretching the question, but it's a great question. But building off of that, in our CAST tools analysis, comparing the sequence divergence of vision versus non-vision, we actually are doing runs where we either include octopus in that or exclude it because of its unusual placement here. So trying to get a sense of, what might be going on. Yeah, Rick?
So, how does your bifocus analysis deal with pseudogenes?
Ooh, that's a great question. I guess it would depend on how divergent they are. So, I mean, the very simplistic answer to that, the real just straight shot one, would be if they are still have enough of that heritage in them to sort evolutionarily into a clade, the pseudogenes will be present. If they have mutated so much that they are basically, by the evolutionary model, no longer recognized as a relative, they will not be. As an example of that, it is, I think, pretty well established that humans have a number of V2R pheromone detector pseudogenes, right? We don't have any functional V2R pheromone detectors, but I'm pretty positive I've read that we have several pseudogenes of them. I think I'd have to go back to be sure, but I'm pretty sure in my phylogeny, no human sequences appeared in the V2R clade. So based on that, it would seem like pseudogenes maybe often are excluded. But it ultimately would depend on how much they've diverged and how the evolutionary model interprets that. Oh, yes.
Yeah, I got a kind of weird question here. So for something like this, like PD 6A, 6C, 6B.
Yep.
I guess we call them duplication events because they seem like they were PDE6 in nature. How far evolved does it have to get to where you'd say they're their own unique gene rather than a six? Why isn't that PD13, for example?
Right. I'll be blunt. My sort of view on this is at times it seems very subjective. There is, I think, to some degree, I think at the end of the day, the truest answer is it's in part subjective. But my impression is that generally, if they seem recognizable as that particular gene, we tend to call them, say, a duplication. I mean, at some point, many genes, if you go way back to the origins of life on Earth, many genes are duplicates at some point in their history, right? I mean, even this entire PDE tree, all 11 PDE families are all related. So they're all really anciently speaking duplications of each other. So I guess what I'm trying to say is an individual gene name tends to denote enough change that we see a distinct identity. It's kind of like the same thinking where you say, well, it's the difference between a species and a subspecies. When does a subspecies become its own species? It's that sort of nomenclature kind of thing. Yeah. Except, you can't ask whether the genes reproduce. So, you lose tools like that, I suppose.
So, Chris, just
Oh,
a
yes.
comment on that is, if you introduce additional characteristics besides the sequence,
Mhm.
it can also help with functional classification. For example, in this one, PD6C is only found in cone photoreceptor cells.
Mhm.
PD6A and B are only found in rod photoreceptor cells. So the cellular basis is part of the reason why it makes sense to divide this gene family into three.
Okay. So this is something too where this is where I often appreciate having Rick along as well, because like Rick always brings like the really great like sort of physiological and like cellular viewpoint. Like a lot of our past talks, you often bring that to the table, and I often tend not to think of it, to be honest. Because like similar function, physiological location, like think of like developmental biology and like embryogenesis. That's a common thing for assessing like homology as well. As how did things originate? What did they evolve from or develop from? I will say I would put out there too, I think it can be a little tricky because of course you also have convergent evolution where things could have like a similar functional basis and appear shared either in like physiology or in biological function, but not be evolutionarily related. So there are some instances like not to go off too much, but like with like Hox genes, for example, in say fruit flies, two of the Hox genes are no longer Hox genes in fruit flies. They are totally different. I think they're considered like maternal effect genes. And you wouldn't even think they're Hox genes, but if you look at them phylogenetically and through other sequence analyses, they are. So I do agree. I think physiology is like super important. That's a context that's really great to have along. But you do also have to be careful not to fall into the trap of like convergent evolution. But yes.
This may have been inferred, and as a parasitologist, I totally missed it. But isn't the octopus eye an example of convergent evolution? And these PDE6s are in octopus eyeballs? And does that explain this apparent convergent?
What's that?
No, they're not.
Oh, okay. The vision part, I'll admit, I'm a little fuzzy on, though it's a big thing for like Dave's focus. But yeah, he says they're not. So
Yeah, that's kind of the most interesting thing is that we know it's a totally different system. So, but they could be doing some other function like maybe controlling the melanosomes or something from melanocytes.
And it could be too, in part of their evolutionary changes here, some of those changes are conserved, but then key functional components are not, right? I shouldn't keep referencing Hox genes, but I can think of some of those where it's a similar thing, like some of the fruit fly ones I mentioned that are no longer Hox genes, but ancestrally are, they lost one domain. So they lost one domain and then everything functionally changed for them. So it could be something like that happening here in some sense, maybe. I'm just guessing at this point. Yeah. Let me just check here really quick. Let's see. Hello. I see someone, Brendan saying mayflies. He's referencing my master's at Plymouth. No relevant questions. Any relevant questions in person?
Yeah, any other questions? And
All right.
if not, we'll get to the next part.
Start cracking your knuckles. All right. Oh, thank you again, everyone. My parents wanted me to make the point. Please feel free to take any snacks you would like. They actually brought some little Ziploc bags. You could take a little to go with you. Thank you all for coming. I appreciate it. Online folks, again, really appreciate you being here. Thank you. I'll talk to you on text.
Great. I do great work, Chris.
Thank you very much. I appreciate it. See ya.
Maybe if you need a couple minutes.
Thank you very much.
He also texted me. He said he wants to know where the horseshoe crabs are.
I started like.
Yeah.
He texted me about the mayflies first.
Yeah, I mean, I'm not surprised. I'm really not.
Asking where mayflies are on the tree. Say Tyler asked.
I actually, well, the thing is, there's no mayfly genomes really that are like well annotated. So,
But yeah, T Money wants to know where the horseshoe crabs are on the tree.
oh, that's one of our other master students at Plymouth from back in the day.
I assumed, but
Wait, was he talking to him too? I didn't send him a link. Well, thank you for passing on the message. I appreciate it.
yeah, I was like, you better hop on. We're taking bets on if you're gonna be on. He's like, I'm gonna be there.
That's nice. Thank you.
Great job.
Yeah.
With the defense part.
Yeah. Maybe I should, folks online, I'm just going to end the live stream now so you don't have to listen to me keep rambling. Thank you all again for being here. I'm just going to stop this if I can. Hi. Oh, thank you. I really appreciate it. Thanks for popping in.
Oh yeah.
Thank you. Thank you. Thank you. End stream. Okay, there we go. I wanted to ask you, when do you think you'll like